I'd like to introduce Walter Wynn, who will get us going with our presentation for today. Thank you.
Hi, I'm Walter Wynn. I live here in Oak Hammock. As you can see, I'm not in the Oak Room today. And my job as facilitator was really easy because Andrea Cunningham spoke a couple of years ago when we were doing this at Oak Hammock, and I asked her if she would be my recruiter, and she said she would. And so all of these people that we'll be hearing in this session have been rounded up. Their arms have been twisted by Andrea. So I will let her introduce our speaker today.
Thank you for the introduction, Walter. Good morning, everyone. I have the pleasure of introducing our speaker this morning, Edward Quinn. So Edward comes from Madison, Wisconsin, and he received his bachelor's degree from the University of Wisconsin-La Crosse in 2011, and is now a PhD student in the UF Department of Anthropology. His dissertation topic studies how psychosocial stress becomes embodied through pathways in the immune system and in the epigenetic regulation of gene expression. And after he receives his PhD in 2023, Edward hopes to build on his interests in public health inequities by working for a government health agency. So Edward, we really appreciate you being here today, and we're looking forward to hearing more about your research. So you can go ahead and start your presentation whenever you're ready.
Awesome. Thank you so much for the introduction. Let me go ahead and start this. Good morning, everyone. Thanks for attending and being here, and thanks again for the invitation to speak. So my presentation is about finding traces of early life experiences in your genes. So let me start off with an unflattering picture of myself at 1 month old. You can probably guess that I never got many compliments on this baby picture. And if you look closer, you can see my left eye is very entertained there. But you can see that I'm a relatively large baby. When I was born, I was 9 pounds and 10 ounces. So definitely one of the larger babies born that day, probably. And right around the time I was born in 1989, there was a man in the UK by the name of David Barker, who was doing some really interesting research related to birth weight and its relationship with chronic diseases in later life. So just a little background on David Barker. As I said, he was a physician and epidemiologist, and one of the things he studied in grad school was early life exposures and their relationship to intelligence in later life. And when I say early life, I mean the earliest life. So he would look at exposures that pregnant women had and think about them sort of as exposures of the developing baby in the womb, and then look at them as sort of predictors of intelligence later in life. So he has this interest in the earliest life experiences in utero, in the womb, during gestation. So throughout his career, as a physician and epidemiologist, he was well aware that there were some changes happening during his lifetime in terms of the kinds of diseases that were becoming more and less prevalent. Right, so he noted, along with many others, that cardiovascular diseases were increasing in prevalence during his lifetime. And given his sort of background research interests, he wondered if there wasn't a relationship between early life experiences in utero and your risk for cardiovascular disease. So he was the first person to look at this kind of question, and in his first sort of pass at trying to see if there was a relationship between those two things, he looked at 212 different sort of localities or districts in England and Wales, and he looked at two things. So one was neonatal mortality. So this is the estimate of the number of infants that are dying in a place in their first month of life. And he looked at that in 1911 to 1915. And then he looked at the same place, the same district locality, let's see, 6 decades later. And what he saw was a relationship between coronary heart disease in that same location 6 decades later with neonatal mortality, or the number of infants that weren't making it past the first month of life. And you can see there's a positive relationship here. And one thing that Barker knew about this measure down here is that this was related to the size of the baby at birth, the baby's birth weight, right? So a lot of these babies that weren't making it through the first month of life were very small. And so that's why I immediately told you my birth weight at the start of the talk. So Barker saw this pattern and he was wondering, is this related to birth weight? And what I'm showing you here is results from a study he did, again, in the late 1980s, where he looked at birth weight, which is listed here. He's giving you categories of less than or equal to 5.5 pounds, less than 6.5, 7.5, and so on. So birth weight is increasing as we move down here. And then over here, what he's showing you is basically your risk of coronary heart disease. And what you can see is that it's going down, it's decreasing, right? And so he's suggesting there's a relationship here, right? Between your weight when you're born and then cardiovascular disease, which occurs, 5, 6, 7 decades later. So that led him to propose this hypothesis, the Barker hypothesis, which this is the way he said it in 2007. Undernutrition in utero and during infancy permanently changes the body's structure, physiology, and metabolism, and leads to coronary heart disease and stroke in adult life. And so that later got expanded to sort of include general early life experiences and general chronic disease, not specifically just coronary heart disease and stroke. But that was the Barker hypothesis. And so that would lead him to, these ideas would lead him to publish articles with titles like this: The Fetal and Infant Origins of Adult Disease, and The Womb May Be More Important Than the Home. And what he's referring to specifically here is that some of his critics would argue that, oh, it's not really about birth weight. Maybe it's about other exposures that people have growing up that are related to coronary heart disease and birth weight. And so that it just looks like birth weight is what's causing coronary heart disease. So normally the way you would address this kind of criticism is you would do an experiment, but Barker was clearly working with humans, and you can't do unethical experiments on humans where, you would purposely say this group of pregnant mothers will have lower weight babies and that group will have higher weight babies. That's unethical, it's impractical. And so it's hard to address those criticisms that, oh, maybe it's not birth weight, maybe, these are just babies from poorer homes, they don't have healthcare access or something like this. And that's what's explaining it. So Barker and others actually had an idea where they could address these kinds of criticisms. And it sort of utilized a tragedy that occurred during World War II called the Dutch Hunger Winter. And what you're looking at here is a picture of a bunch of bags of food that have been dropped in the Netherlands by the Allies in 1945. And the reason they're dropping food in the Netherlands is because the Dutch have just gone through a famine during this winter. So the Nazis were occupying the Netherlands in late 1944, and people thought the Allies were going to be coming really soon. And so some of the Dutch sort of rose up and rebelled against the Nazis. And the Nazis retaliated by cutting off food supplies. And eventually they sort of relented and allowed food to come by boat, by waterways, basically, in the country. But it was winter, and it was a harsh winter, and those waterways froze. And so that cut off another sort of the only source of food, really. And so what you had then was this Dutch hunger winter, this famine, where for sort of very suddenly, nutrition is sort of cut off, and then very suddenly it's sort of turned back on again. And so what Barker realized and other researchers realized is that we could study the mothers who were pregnant during this famine, mothers who became pregnant during this famine, and then compare them to mothers from the same hospital before the famine, after the famine, and see if there were any differences there in their babies and their health outcomes. And it turns out there are a lot of differences. So there's a whole lot of text here. You certainly don't need to read everything. I'll just draw your attention to these few lines here. So here's type 2 diabetes. That's the outcome we're looking at. And then these 3 columns here correspond to when the developing baby was exposed to famine. So either an early, mid, or late gestation. So if you look at an outcome like type 2 diabetes, what they saw was that babies who were exposed to the famine in late gestation had an increased risk of type 2 diabetes compared to babies born before or after the famine. And that's at age 50. At age 50, they can detect a difference between those who were exposed to the famine in utero and those who were not. For coronary heart disease, those who were exposed to the famine in early gestation had an increased risk of coronary heart disease compared to babies who were unexposed. And that was detected, that difference, right? You could see that difference at 50 and 58 years. And this, I mean, there's a lot of outcomes here, and it goes on. There's type 2 diabetes, coronary heart disease, even overall brain volume in men, right? That's reduced in the men that experienced the famine in utero in early gestation compared to people who did not go through the famine. So, what is this showing us? This is showing us that these early life experiences have a relationship with your risk for chronic disease much later in life, and even your brain volume, which is astonishing, I think, when at least it was for me when I heard it the first time. But back to birth weight. So that's an example of a nutritional stressor, right? Where there's less food being consumed by the mother during pregnancy. And so that's a nutritional stress. But there are also examples of other kinds of stressful exposures in utero that have an impact on birth weight, for example. So if we look at the study from 2006, which was really interesting, what they did is they looked at Arabic named women, I believe this was in California, before and after 9/11. And they looked specifically at women who were giving birth. And what they saw, if they compared the 6 months after 9/11 to the same time period a year earlier, is an increased risk in low birth weight, in rates of low birth weight among Arabic named women specifically. And there's all kinds of evidence about how this group of people, Arab Americans, experienced increases in discrimination and a rise in hate crimes following 9/11 because of their identity. and that had an impact on birth weight among people who had Arabic sounding names. Another example of this sort of thing comes from Postville, Iowa. So in 2008, there was like the largest immigration raid to date, with like 900 ICE agents, I think, in like a Black Hawk helicopter surrounding this meat plant. And a lot of people got arrested. And, you know, this was very traumatic for the Latino community, especially in Postville. And so what Novak et al. Did in 2017 is they looked at Latina mothers who gave birth before and after this immigration raid, and in the entire state of Iowa. And what they found was an increase in the rates of low birth weight following the immigration raid among Latina mothers specifically. So this is really interesting stuff, right? There's these really early life exposures. They have an impact on you know, your risk of chronic disease. They impact birth weight. Birth weight might be related to chronic diseases. So how is this happening, right? How can we understand how these very early life experiences impact your birth weight and then your risk for chronic diseases? And like, how is your body remembering, or how is it impacted by these experiences? And so to try and understand that, scientists turn to rats. So what I'm showing you here is a rat mother and her rat pups that are nursing. And I didn't know this before I got into this, but there's variation in I guess how loving rat mothers are. So some rat mothers lick and groom their rat pups often, frequently, a lot. And then there's some that do it less, right? And what's really interesting about this maternal behavior towards these rat pups is that those differences in maternal behavior leave a mark on the DNA of the rat pups. So specifically here, I'm talking about epigenetics, which literally means on top of genetics. And specifically, I'm talking about a kind of epigenetic mark called DNA methylation. And so DNA methylation, here's sort of an illustration of what DNA methylation looks like. So here's your classic double-stranded DNA, and then these golden orbs are sort of like tags on the DNA. And these tags are what we call DNA methylation, and people vary in terms of how much DNA methylation they have in their genome. And what was shown with the rats in the previous picture was that things like natural variation in maternal behavior can impact DNA methylation. And the reason that's important is because DNA methylation impacts how your body is using your genes. Basically, it can help to silence or express some of your genes. And so you have this maternal behavior that's impacting DNA methylation in the rat pups. And what's really interesting is that it's reversible. So if you take the rat pups from a rat mother who licks and grooms a lot, and then you take those rat pups and you move them into a different cage with a mother who doesn't lick and groom as much, then you'll see changes in this DNA methylation. So these early light, and these are within the first week of life for the rat pups. So these have stable effects, you know, throughout their life, and consequences for how they respond to stress later in life. So this is really interesting. This is one way that early life experiences might be getting embedded in your DNA and then sort of shape how you respond to different stressors later, or maybe even affect your risk of chronic diseases, for example. All right, so the natural next question is this kind of thing happening in people, right? Are people becoming exposed to, you know, stressors in early life, and then maybe that's you know, the effect that we see 5 and 6 decades later? How is that happening? Maybe it's through changes in DNA methylation in early life, right? That's a potential explanation, because it's something that regulates your genes. It's sensitive to your environment. It's potentially very stable as well. It could last decades. And so that the question that my advisor here, Dr. Mulligan, and people in our lab, many people in our lab have worked on, is this kind of programming effect, is this change in the DNA related to an exposure in early life stress related to birth weight, for example. And so this work has been done both in my advisor's lab, Connie Mulligan, and with a collaborator in the psychology department, Dr. Darlene Kirtis. The place where this research is done, and the group of people that we look at these kinds of questions of early life stress and DNA and birth weight is in a city called Goma, which is in the Democratic Republic of Congo. And so here's DRC, Democratic Republic of Congo, in Africa. And Goma is on the border of DRC with Rwanda here. And that becomes important, actually, because I'm sure as many of you have probably heard in the past, there has been some violent conflict and genocide in Rwanda. And sometimes that conflict has spilled over and continues to sort of spill over into Congo and in this region around Goma. So this is a city of around 2 million people. It's on the eastern border of Congo. And some of that spillover effect I was talking about from Rwanda results in armed groups battling in and around the city in very recent times. Like as recently as 2011, a rebel group had taken control of the city. Another consequence of this conflict is that rape has been used as a weapon by these armed groups to try and subdue the people who live here. So there's very high prevalence of major depressive disorder and PTSD in this region. And so this is a very stressful, difficult place to live, especially for women. So back in around 2008, I think it was, 2009, my advisor, Dr. Connie Mulligan, and a graduate student in the program, Nicole Rodney, started a collaboration with this hospital, Heal Africa in Goma, which is pictured here. And Heal Africa is a holistic hospital specializing in women's health. And they also offer treatment for victims of sexual violence here. So they help women recover and also try to teach them skills to be able to earn money and things like this. So here are collaborators from Heal Africa Hospital who have worked with us on the projects that I'll describe shortly, to look at this question of early life experiences and whether DNA methylation might be related to your later life health and birth weight and things like that. So these two women here, Anne-Marie and Chantel, they interview all of the mothers that are recruited at the hospital to participate in the research projects about their stressful life experiences. This is Georgette. She's a midwife that works at Heal Africa. This is Bichot. He's the lab manager who helps to process samples that are collected during the interviews. And this is Felicienne. He's kind of a jack of all trades. Keeps the wheels on the bus. And Felicienne was a nurse at Heal Africa Hospital. And then after this project was started, he actually came to UF and got his PhD and graduated last year. And he's back in Goma now, and he's doing research there. So that's nice. It's a continuing relationship. So the basic conceptual model of the work that has been done, at Heal Africa Hospital with these mothers in Goma, is that maternal stress is going to impact newborn outcomes, such as birth weight. And so what we're interested in testing is whether DNA methylation, that little tag on the DNA that I showed you earlier, whether that is playing a role in sort of connecting these two things, connecting maternal stress and newborn birth weight, for example. And that could be a way sort of that the body is programmed in a particular way or put on a particular developmental trajectory that could explain why these early life experiences have impacts 5 and 6 decades later. Right, and so that's sort of what we're getting at in the end there is that this is probably related to chronic diseases later in life. And there's a lot going on here, but you certainly don't need to read everything. The point of this slide is just that in an initial small pilot study, what my advisor and other members of the lab found was that indeed there are relationships between the methylation of a newborn baby, the DNA methylation, the tags on the DNA, and the mother's experiences of stress related to war. There are also relationships between the baby's DNA methylation and their birth weight as well. So this was some initial support for this model, right? This idea that maternal stress might be related to DNA methylation, which might be related to newborn outcomes. So as I said before, we've continued to collaborate. That study was published in 2012. But this was looking at DNA methylation in one region of one gene. And we've all got 20,000 genes in all of our cells. So there are a lot of other places to explore, right? Because there are 28 million places in your genome where you could have or not have DNA methylation occurring within a single cell. So what about other parts of the genome or the genome as a whole? If we could get some kind of summary measure of the DNA methylation tags that are there, that would be useful, right? And what about follow-ups? Like what happens to the DNA as these infants start to develop into children? So these are questions we're looking at now, and in order to do that, we're following the children over time. So all I'm showing you here is that we've recruited mothers and their newborns at birth, and we followed them at 6 months. Each dot represents an interview and a sample collection, so we could look at the DNA of these children at 6 months, at 1 year, at 2 years, and then at 3 years. So we can kind of ask not just sort of what's happening at the moment at birth, basically, but what happens as these kids start to develop. And I mentioned that the initial study on this question was on one region of one gene, right? and that maybe it would be helpful to have a measure of DNA methylation that's more sort of global, right? So one measure that we can use is called DNA methylation age. So let me just talk about this one for a second. So as we age, there are changes in our DNA methylation that occur naturally. So at some places in the genome, you get more methylated as you grow older. In other places in the genome, there's less methylation as you grow older. And what was discovered in 2013, actually simultaneously by a few different people, is that those changes in your DNA methylation could be used to create a measure that predicts how old you are. So we could look at the DNA, at your DNA. We can look at the methylation at these 353 different places. And then we could spit out a guess of how old you are without you telling me how old you are, right? And what we see is that if we take that guess of your age from the DNA methylation, which is down here, and we compare it to your actual age, what we see is that there's a very strong correlation. There's a positive relationship here, right? So basically this measure using DNA methylation of your biological age, we might call it, your epigenetic or your DNA methylation age, is very closely related to your chronological age. As far as I know, there's no other biological marker that is so good at guessing what your age is as the DNA methylation age is. But so that's fascinating in itself, and people have extended this to like horses and monkeys and rats and all kinds of other animals. But I think what's more interesting even is that if you take the chronological age and we put it on the x-axis this time, and your DNA methylation age, and then we draw a line sort of describing the relationship between these two things, basically trying to predict your DNA methylation age with your chronological age. You can see that some of the dots are above the line, like this one is above that line, and the line represents our best guess for your DNA methylation age based on your chronological age. Some people are above the line, right? Some people are below the line. And it turns out that if you're above the line or you're below the line is really useful information, right? So that difference between where your DNA methylation age is and where your chronological age is turns out to be predictive of all kinds of things. And I'm just showing you a few of them here. So we're looking at cancer and COPD and cardiovascular disease. And what these stars are telling you here is that if you fall above the line on this graph, right, you're at an increased risk of cancer, for example, or cardiovascular disease. Or COPD there, for example. So that measure, right, this distance between where your DNA methylation age is and where your chronological age, we refer to that as DNA methylation age acceleration if you're above the line, and deceleration if you're below the line. And most of that work has relating that difference to cancer and cardiovascular disease has been performed with adults. But one question that a few people have started asking is, well, what does that difference mean in kids? And so here are some examples. Simkin et al. In 2016 found that birth weight was related to age acceleration, so falling above that line in 7-year-olds. Jovanovic et al. Found that exposure to violence in kids aged 6 to 11 was related to age acceleration, so again, falling above that line. And then the same thing with the amount of stress hormones in the body in children, that is related to increased DNA methylation age acceleration, falling above that line. And so this is something I wanted to look at in my research in the Congo, where we're following these kids in their early years of life. And so if we plot how old the children are here on the x-axis relative to their DNA methylation age, you can see it's going up as expected. And if we draw a line through here and we think about, okay, who's above the line, who's below the line, right? Who has DNA methylation age acceleration and who has DNA methylation age deceleration, right? Because we know that this is important for predicting your risk of chronic disease, for example. So if we look at that difference between where you are relative to the line and we plot that difference, this is what it looks like. So if you're above 0 here, that means you have DNA methylation age acceleration. And if you're below 0, that means you have DNA methylation age deceleration, right? And again, acceleration is generally associated with higher risk of chronic diseases in later life. So I guess, one of the things you can notice right away is that everyone's pretty tightly clustered around here. This turns out to be an outlier. Pretty tightly clustered around 0, maybe just below on average at birth. But then as we move on, to 1-year-old, 2-year-olds, and 3-year-olds, there's a lot more spread in this DNA methylation age acceleration measure. So the question is, who are the children that are ending up on this top half of the graph? And who are the children that are ending up on this bottom half of the graph, right? Like, what is predicting whether as a child you're going to have DNA methylation age acceleration or deceleration? So one pattern I wanted to look at right away was sex, because it's well known in adults that males have methylation age acceleration, higher levels of it relative to females generally. And this corresponds to life expectancy even, right? We know that males typically live shorter lives than females. And typically males have higher age acceleration than females in adults. But if we look at the graph here, there's not sort of an immediate pattern, right? It's not like all the red dots symboling males are all above the line. All the blue dots symbolizing females have deceleration. That doesn't appear to be a pattern here. But if we look at birth weight, and what I'm showing you here is the distribution of birth weight in grams. And so if I draw a line here at 2,500 grams, that's the World Health Organization cutoff for low birth weight, where doctors will start to take into account that you're low birth weight and maybe this will affect your healthcare treatment from birth onwards. And so if I draw a line there and I split up the children who are participants in the study into low birth weight and then high birth weight babies, and I highlight the DNA methylation age acceleration by this, it's still kind of hard to see any sort of pattern. It really doesn't look like much, except for maybe at 3 years, you can see all the low birth weight babies are way up here. There's only 4 of them though, so it's hard to say if that's really a trend. But if we use statistical models and control for things like gestational age, and we can control for sex, and then we can model sort of the trajectory of DNA methylation age acceleration or deceleration according to whether the baby is low or high birth weight. And if we do that, this is what we see, where in the red, we see low birth weight babies show a trajectory of DNA methylation age acceleration, right? They're above 0 here. So their DNA methylation age is older than we would expect based on their chronological age alone. And then if we look at babies in the other group of birth weight, the high birth weight group, we can see that their trajectory is one of DNA methylation age deceleration. And so that's really interesting because we know that DNA methylation age acceleration is associated with cancer and cardiovascular disease. And what we're seeing here is that your birth weight, which is also related to cardiovascular disease and risk of diabetes, is predicting your DNA methylation, your epigenome, basically. These changes in DNA methylation over time that are related to your risk of chronic diseases. So this is a possible explanation for how these early life experiences might be related to your later life risk for diseases. Which this is kind of just what I said. So the idea here is that early life experiences, right, might shape your birth weight through these changes in DNA methylation. And then we can use that as a marker, birth weight, of your early life experiences and look at the trajectory of your epigenetic aging and see that this might be one way that we can link birth weight and later life risk of chronic disease is by looking at birth weight as a marker of those early life experiences and then using that to sort of see how those early life experiences are affecting the trajectory of your epigenetic aging, which then is related to chronic diseases later in life. So I just showed you results from one sort of DNA methylation or epigenetic clock, but there are a lot of clocks out there. It's like there's a new one published every day. So I could potentially look at some other clocks that are trained in different ways. And also in the figure I showed you, I wasn't incorporating any maternal factors, so those might be important to include as well, like delivery method or age or the size of the mother, for example. So here are some references, and I would like to acknowledge the women and children participants in GOMA at Heal Africa. You know, they are the ones that made the study possible, as well as the collaborators at Heal Africa and Dr. Darlene Kirtis at UF Psychology. So funding for the research was provided by the National Science Foundation to Dr. Mulligan and Dr. Kirtis. And funding for my graduate studies was provided by the UF Graduate School. So that's all I have. I'm happy to take some questions if anyone has any questions about stuff that I talked about or clarifications, or maybe I just lost everyone immediately. Who knows?
Ron?
Hi,
Yeah,
Ron.
I'm just wondering, with what you're finding, would you envision perhaps a decade from now that doctors would begin early when they feel they have someone who's above the line, a baby above the line, that they would begin instituting some kind of preventive care, or anticipatory care. Do you think we could come to that?
Yeah, that's a great question. Sort of what are the clinical implications, and could these be used in clinical practice? I would bet that there are patent applications, if not already filed, like they're getting filed right now, and people are gonna widely implement a measure like this to look at that. I think so. I think we're still sort of trying to understand what the measure means in children, because there is some conflicting evidence. So sometimes a stressor might be related to being above that line. Sometimes in other cases, it means you're below the line. So it's not entirely clear what this measure actually means in children. But I definitely see it impacting clinical practice in the future, for sure.
Thanks.
Yeah.
Go ahead, Walter. Walter, go ahead.
Yes, thank you. That was a very, very interesting talk. I'm an engineer, so I love graphs. And your graphs are quite interesting, but my question is related to this, the DNA methylation. You indicated sort of that it was repairable. And my question is, and it's probably too early to know, is it hereditary? If I have some broken genes here, have I passed them on to my children and grandchildren? And what does that mean to them and to me?
Yeah, that's a great question. Certainly, DNA methylation is influenced by your actual genetic sequence. That is an influence on it, especially at birth. We see that there's a large heritability, but that as time goes on, that as you live your life, as you have more experiences, your DNA methylation is responsive to that. So the relationship between your mother's DNA methylation and your own, might diverge over time. Oh, is there a question in the chat? Oh, okay. Oh, I see. Oh, it's Andrew. All right. Hopefully it's just a test. Great questions. Anyone else have a question?
I don't see any other questions, but Walter, do you want to make any final comments, or Andriana? Or I guess you can't unmute now.
Yeah, once again to Andrea, fantastic selection of people. These have been very, very interesting. And we are going to skip a week. And the next one will be August 16th with Rose Bryson, PhD student in biological anthropology and an analyst at a forensic anthropology lab in Gainesville. So maybe we'll have some cop talk or some detective work or whatever. Andrea, add more to it, please.
I don't think she wants to unmute right now. Well, thank you everyone for being with us, and I'll send out a reminder that we will not have a presentation next week, but everyone continue to stay well, and we'll see you in 2 weeks. Thank you again, Edward.
Thank you. Thanks for listening. Thanks for the questions.